Hi, let's talk about an application of intermolecular forces inside of biological systems. So IMF, intermolecular forces, this is going to be those covalent compounds and how the different compounds attract to one another. You're thinking hydrogen bonding, dipole-dipole, London dispersion forces. How those relate to the structure and function of biological processes. Now, definitely want to give credit to AP. This is coming from the AP chemistry framework. If you're in AP chemistry, this is something that's been published. If AP took the time to put the ink to paper, even though I have not seen a lot of this on the AP test, I think it's good for you to know. And if you need examples for your class of where intermolecular forces contextually where they exist and to give examples of IMF, these are all outstanding examples. So a substrate interaction with an active site of an enzyme. I drew a picture of this. So this is just catalysis inside of the body. Where a chemical reaction will happen faster because of the enzyme decreasing the activation energy, and it all depends on intermolecular forces. So this would be your enzyme right here. This is the active site. It kind of looks like Pac-Man for those of you who know what I'm talking about, that little chomper. And then this would be your substrate. So for those to come in and bind, the enzyme will have the chemical reaction for that substrate go faster. But in order for those to come together like this, it's going to be intermolecular forces that attract them. So you might have like polar sites right here and non-polar sites right there. Remember that like attracts like. So polar and polar will be attracted, non-polar will be attracted. Likewise, hydrogen bonding will interact, whereas LDF, that will interact. Okay, next, the hydrophilic and the hydrophobic regions of proteins. I like the example of our lipid bilayer, whether it be inside of a cell, or I should say encasing around the cell, where you have the hydrophilic, so this is going to be water loving, and a hydrophobic, water hating, if you will, and it's the fatty acids that are in the middle. Those are going to be the hydrophobic, and it's the outside that you have the polar ends. They're going to be the hydrophilic, and that's what surrounds the cell. So really good example of intermolecular forces. Non-polar going with non-polar, and then the polar going on the outside. Large biomolecules, and I'm going to give DNA as the example for this. This is the interaction between different regions of large molecules, and DNA, way cool. So you know, in DNA, there's a sugar, a phosphate, and then the acid, the nucleic acid that's attached to that. And if you look at the outside edges of DNA, so pretend we have this helix, there's no specificity on the outside. It's just sugar, phosphate, sugar, phosphate, sugar, phosphate. The specificity is on the inside. It's the rungs of the DNA. So cool. And you'll recall adenine is attracted to thymine, and guanine is attracted to the cytosine. Guess how they attract? Intermolecular forces, hydrogen bonding. The A and the T are held together, attracted by 2 hydrogen bonds, and the G and C are attracted, held together by 3 hydrogen bonds. Now remember, hydrogen bond is really not a bond, it's just a force. So yes, the backbone of DNA, you have those, what's it called, the dipeptide bond, that I believe that's what it is, that holds the sugar phosphate, holds all of those together. But in the middle, the rungs, there's not actually an intramolecular force. What holds those together is simply an intermolecular force. It's an attraction between the hydrogen bonding of A and the hydrogen bonding of T. I just think that's fascinating, really cool. So powerful, all of our DNA, intermolecular forces, neat. Next, function and properties of molecules depend on the shape, which is dictated by their intermolecular force interaction. So great example is coming back to our enzyme and substrate again. You know that proteins fold in a tertiary, a 3D pattern, and they have to be super specific. If they do not fold correctly, it's over. They won't work. Or if they're denatured and they go back into that straight line, is that called the secondary? I think, I'm not sure. Forgive me, my biology students. It goes back into this string of proteins. It won't work. So that folding, the proteins fold, and you can have huge, huge molar masses, 50,000 grams per mole on the molar mass on some big protein. It has to fold just perfect. How does it fold? Intermolecular forces, non-polar regions touching each other, polar regions touching each other. So pretty powerful, really, really neat. Next, properties of synthetic polymers. So synthetic, this means man-made, something that we made, like a synthroid, where yes, we can find this in nature, but it might be costly or difficult, so we'll make the same thing. So synthetic polymers, they're modified by change in the chemical composition and the structure. So how can we modify them? How can we change them? We're thinking those intermolecular forces. And then lastly, the function of the enzyme is dictated by its structure, absolutely. And that goes back to a protein folding in that very specific 3D pattern so that it has polarity and non-polarity certain areas so that there can be this interaction between the substrate and the enzyme. So the shape, very, very important, which is held together by intermolecular forces. So all throughout life, biological processes, wow, huge amount of intermolecular forces, which is pretty neat. Well, I hope that helped you. Have a great day. You're doing good. Thanks.